All right, let's do something a little different today. We're going to shrink ourselves down, way, way down, for a safari into a hidden world. We're going to explore one of the most successful life forms on our planet, bacteria. These guys are the ancient, invisible architects of our world, and we're about to take a guided tour of one of their cells. So, let's start with a number just to set the stage. 30 trillion. That's the estimated number of human cells that make you, well, you. It's a mind-boggling number, right? It's everything from your brain to the tips of your toes. But here's the wild part. In and on your own body, you are hosting even more bacterial cells. Yeah, you heard that right. You're not just a person. You're basically a walking, talking ecosystem. So if we're outnumbered in our own house, it's probably a good idea to get to know the neighbors, don't you think? Okay, so to really get a handle on bacteria, we first need to figure out where they fit in the grand tree of life. And let me tell you, they're not just some branch on the tree, they're pretty much the roots. Every bacterium you've ever heard of belongs to this massive ancient family called Kingdom Monera. I mean, these are the original inhabitants of Earth. They're most abundant organisms out there, and they are all single-celled life forms called prokaryotes. And that one word, prokaryote, is the key to understanding just how different they are from us. And here's that core difference, side by side. Over on the right, you've got our cells, eukaryotic cells. See how they're all complex and organized, with that nice, neat nucleus in the middle to hold all our DNA? Now look to the left. That's the prokaryotic cell, the bacterial way of doing things. It's much simpler. Its genetic material isn't tucked away. It's just kind of floating around in a specific area. And that area has a name, the nucleoid. Think of it like a command center, but a command center with an open floor plan. There are no walls, no corner office. It's just this dense, tangled-up region where all the genetic blueprints are kept. And this elegant simplicity, well, it's one of the secrets to their incredible success and their ability to adapt so quickly. So now that we've got the basics down, it's time to start our tour of the bacterial cell. We're going to peel back the layers one by one, from the outside in. We'll explore all the essential parts and uncover what makes these little machines tick. Our tour is going to have 3 main stops. First, we'll look at the external stuff, the tools bacteria use to move around and grab onto things. Then, we'll dig into the cell envelope, its tough, protective shell. And finally, we'll venture deep inside to check out the machinery that keeps the whole operation running. Let's kick things off with what is arguably the most critical piece of bacterial hardware, the cell wall. This isn't just a wall, it's the bacterium's fortress. And its design is so important that it lets us split almost all bacteria into one of 2 major groups. And trust me, this isn't just for textbooks. It literally has life or death implications. So, the secret ingredient that makes this fortress so strong is a molecule called peptidoglycan. Now, I know, that sounds super complicated, but just picture it as a kind of molecular chainmail. It's this mesh of sugars and proteins all linked together, giving the cell its shape and, crucially, stopping it from bursting under pressure. But, and this is a huge but, not all bacteria build their chainmail the same way. On the left, you've got gram-positive bacteria. They wear a really thick, heavy-duty coat of peptidoglycan. But on the right, you've got gram-negative bacteria. Their layer of chainmail is thin, but they add something extra, a whole other outer membrane on top of it. This one difference in architecture is a total game changer. And this table really breaks it down. Gram-positives, thick wall. That's why they hold on to that classic purple gram stain. Gram-negatives, thin wall, plus that extra outer membrane. Now, that outer layer is a huge deal. It contains a substance called lipopolysaccharide, or LPS for short, which can be toxic to us. But even more importantly, that outer membrane acts like a selective barrier, making it way harder for some of our best antibiotics to get inside and do their job. It's a big reason why gram-negative infections can be so tough to treat. And this whole idea, this fundamental split, all comes back to a simple staining test, developed way back in 1882 by Hans Christian Gram. And get this, over 140 years later, the Gram stain is still one of the very first, most important steps we use in the lab to figure out what kind of bacteria we're dealing with and how to fight it. All right, we've inspected the fortress walls. They're impressive. Now, let's zoom out a bit and look at the tools and gadgets bacteria have bolted to the outside of that fortress to help them survive out in the world. Yeah, beyond just the wall, bacteria can be kitted out with an incredible array of different appendages. And these aren't just for looks. Each one is a specialized tool for moving, for sticking to things, and even for communicating. First up, for getting around, many bacteria have flagella. These things are amazing. They're basically tiny biological outboard motors, long, whip-like propellers that can spin around and drive the cell through liquids. This lets them actively swim towards stuff they like food, or get away from things they don't, like toxins. So what if a bacterium doesn't want to move? What if it wants to stay put? For that, it uses things called fimbriae and pili. Fimbriae are like tiny little grappling hooks, these short bristles that help the cells stick to surfaces. Pili are a bit longer, and some of them have a very, very cool job. They can connect to bacteria and transfer bits of genetic information between them. This is a primary way they share useful traits, like, you guessed it, antibiotic resistance. And for the ultimate defensive upgrade, some of the more dangerous bacteria have a capsule. You can think of this as a slimy, sugary invisibility cloak. It makes the bacterium super slippery and hard for our immune cells to get a grip on, so they can't be gobbled up. It's a clever trick that gives them a huge advantage when causing an infection. Okay, we've explored the armor, we've seen the weapons, and we've checked out the tools. For the final stop on our tour, let's slip past all those defenses and take a peek at the essential machinery that's running the whole show on the inside. So inside, you find the basics for life. You've got the nucleoid with all the DNA, and you've got the ribosomes, which are little factories for building proteins. But some bacteria have an almost sci-fi survival tool called an endospore. When things get really tough, no food, extreme heat, whatever, they can form this tiny, dormant, armored pod. This is beyond hibernation. It's like a state of suspended animation. They become incredibly resistant to heat, radiation, and disinfectants, allowing them to just wait, sometimes for centuries, until conditions get better. So when you put it all together, from their simple, efficient design on the inside to their complex defenses on the outside, and their just unbelievable resilience, bacteria are the undisputed masters of survival. They've been on this planet for billions of years, adapting and evolving in ways we're still just beginning to wrap our heads around. It really makes you wonder, doesn't it? As ancient as life itself, what can these tiny survival machines still teach us about life, about adaptation, and about our own place in the world?